And then it's control alt laser.
Okay. Hi, everyone. I'm really happy today to introduce Julia Gershon. Julia is a sophomore here at Stanford, moving on to junior year, right? Julia started working in the relab as an intern last fall, and we were for a couple hours a week. And we were lucky enough to have her come on full time as a summer intern this year. She was working in the relab with both myself and Purva. And with Purva, she was working on cloning kind of a ridiculous number of genes from sorghum. I think the pipeline included something like 150 gene candidates. So between balancing that and working with me on a project studying the metabolic burden of an organelle and cyanobacteria under different environmental conditions, she's kept very, very busy and has produced an outstanding amount of work. And so I'm going to clear out and let her present what she's been working on. So please join me in welcoming you.
Thank you so much. So today I'm going to be talking about when carbon concentrating mechanisms, which are usually used to aid photosynthesis, can actually be a bad thing. Specifically, I'd like to talk about this in cyanobacteria. Cyanobacteria are a good organism to explore this because they are really great at photosynthesis. Despite making up only 0.2% of photosynthetic biomass, that's this tiny stripe on this inner pie chart here, they account for more than 35% of the world's global carbon dioxide fixation. And one of the reasons that they're so good at photosynthesis is because of their carbon concentrating mechanism. So many organisms develop carbon concentrating mechanisms to improve their photosynthetic activity. These things work by keeping carbon concentrated around Rubisco so that it can function better, which leads to better photosynthesis. And the cyanobacteria's carbon concentrating mechanism is called a carboxysome. It's essentially a compartment within the cell that keeps Rubisco inside it, as well as carbon dioxide, and it keeps the oxygen that is diffused throughout the rest of the cell out. Now, this is important because while carbon dioxide is catalyzed by Rubisco for photosynthesis, Rubisco can also catalyze oxygen for photorespiration, which is wasteful. So when the Rubisco is kept separate from the oxygen, the cyanobacteria are able to really maximize their photosynthesis without worrying about photorespiration. However, to explore the impact of the cyanobacterial carboxysome on a cell, a mutant strain was created without a carboxysome called the delta CCM. In this strain, Rubisco is diffused throughout the cell with all of the oxygen, which means that carbon dioxide must be competing with oxygen for being catalyzed by Rubisco. This means that it's a little bit worse at, or a lot worse at photosynthesis than the wild type, and it has higher photorespiratory activity. So as a result, the delta CCM performs a lot worse in air. These here are some images from cyanobacteria cultures grown on plates with wild type and delta CCM in atmospheric carbon dioxide. So that's about 0.04% carbon dioxide. And you can see that the wild type grows quite happily while the delta CCM doesn't. We were able to replicate these results in a liquid culture and get this. So on your y-axis here, we have the optical density, which is a good indication of culture density and culture growth. And you can see that while the delta CCM in red shows basically no growth in air, the wild type grows quite happily. So clearly the carboxysome gives the wild type a fitness advantage in low levels of carbon dioxide. However, when the levels of carbon dioxide are increased and carbon is no longer a limiting factor, the delta CCM performs a little bit better than it did before. It's essentially equal to the wild type at high or 3% carbon dioxide. And again, we were able to recreate these in a liquid culture and see that the optical densities grow at about the same rate. So we've established that the wild type has an advantage over the delta CCM in low carbon dioxide environments, and that the two strains can perform equally in high carbon dioxide environments. But so far, there's been no record in the literature of the delta CCM performing better than the wild type. And this might not come as a surprise to very many people because the carboxysome is a really helpful tool for photosynthesis. So removing it might not seem like something that would give a fitness advantage. But I'd like to point out that cyanobacteria grow in a lot of very diverse environments. This picture on the left here is of a carbonated spring in Colorado where cyanobacteria grow, and it has levels of carbon dioxide in the water high enough to support growth of the delta CCM. So in scenarios like these, the carboxysome is no longer helping the wild type, as the delta CCM does just fine without it, but the wild type can't get rid of it. It's sinking energy and resources into this thing that it can't get rid of and isn't healthy. So in these sorts of situations, we would hypothesize that the carboxysome is only a hindrance and might actually be detrimental to the wild type. However, that being said, so far, there's no record in the literature of this being found. But I would argue that the literature is, most of the research that has been done on these strains has been done in very specific scenarios. The cells are being spoon-fed all the resources they need and being kept mostly in log phase. This isn't really representative of how they grow in nature or even in industrial settings. So I have some photos here of the many different places that cyanobacteria grow. We have some algal blooms, rocks on the Tahoe shore, a side of an underwater cave, and an underwater lake in the Antarctic, as well as a bioreactor used to make degradable plastic. So in all of these very varying scenarios, there is one thing that is kept constant, and that is that these cyanobacteria grow very densely, whether that's in mats or blooms or in a reactor where you're trying to maximize product while minimizing input. These cyanobacteria are very resource starved in some cases and are often very dense. So while we know that the delta CCM can perform just as well at high CO2 and that the most representative instances of real world growth here happen in very dense cultures, we wanted to explore how the delta CCM and wild type size up to each other in dense cultures that are experiencing high carbon dioxide. So to imitate this, we took a flask of the wild type and a flask of the delta CCM and put them in an incubator at high carbon dioxide levels and basically didn't touch them for 2 days. We didn't give them any more nutrients or any more media, we just let them grow. And at the end of those 2 days, this is what we found. So these two strains have been grown up for the same exact amount of time in the same exact conditions, and you can see that the wild type looks quite unhappy. It's bleaching, it's yellow, it's lost a lot of its chlorophyll, while the delta CCM is doing fine. So already this is suggesting that there is something about the cost of maintaining a carboxysome when it's not helping you that is taking a fitness cost on the wild type. The question was, however, would this inhibit growth if we gave the wild type more nutrients to try and continue? So that's what we decided to do. We diluted these cultures down after the wild type was looking unhappy and the delta CCM was looking fine, and we allowed them to regrow. So for context, this was how they grew at the beginning when we first put them in the incubator from a healthy culture. So here you can see the optical densities increase at roughly the same rate. They're both happy and moving along nicely. And then after dilution, when we allowed them to regrow, their rates look like this. So you can see that the delta CCM recovers quite nicely. Its optical density increases pretty quickly. The wild type isn't exhibiting no growth, but it exhibits very, very little growth, suggesting that the early bleaching caused by not having a carboxysome does limit its growth. So this is already really interesting to see in individual strains, but I would go as far as to argue that individual strains are not the best way to explore how these two strains would size up in the real world. A better way to do that would be in a co-culture. In co-cultures, you make sure that the strains are in the exact same conditions, and it's more applicable to the real world because the two strains are fighting for the same resources. So there are many different reasons why the wild type could be bleaching like it is, and some of those could be a result not of performance on the individual cell level, but on the culture. For example, if the wild type blocks more light, it could block out the light from itself, and basically that would be a culture effect that would be negatively impacting the wild type. However, if we put the wild type and the delta CCM in the same culture, then they will both be experiencing that effect if that is the case. Similarly, it could be possible that the wild type is bleaching because it consumes resources faster. However, when they're in a co-culture, if that's the case, it would starve the delta CCM just as much. Similarly, if the wild types are making the media environment more harmful, that would impact the delta CCM too. So by doing this in a co-culture, we can make sure that all of the variables are the exact same because these cells are literally in the same flask, so they're in the same environment. We can understand if the effects that we're examining are on the individual cell level or on a wider culture scale. So to set up our co-culture experiments, we made replicates of 3 different types of cultures. Wild type and delta CCM cultures. These serve as good controls, and when the wild type starts to get yellow, we can tell, which is helpful. And a co-mixture that was a 1-to-1 mixture of the both of them. So we set all of these up starting with equal optical densities and allowed them to grow through their growth curve, and then diluted them back down and allowed them to regrow again. So as we were doing that, we were collecting samples the whole time for further measurement. And we analyzed these samples in a lot of ways. We measured optical density to understand how the cultures were growing. We took lots of pictures. That helps to visually see the culture growth and also to understand how much bleaching is going on. And we measured allele frequency. So when we started this experiment, right before the dilution, you can see that the co-culture seems to be an intermediate between the delta CCM and the wild type. The wild type looks slightly bleachy, the delta CCM is very happy, and the co-culture is somewhere in between them. This makes a lot of sense because we've shown in the past that before dilution, the two strains grow equally, so they should be equally contributing to the co-culture as well. However, after dilution, after oversaturation and dilution, the co-mixture recovers faster than the delta CCM. Now, we already knew that the delta CCM the co-mixture recovers faster than the wild type. We already knew that the delta CCM would recover faster than the wild type, and it makes sense that the co-mixture falls somewhere in between, but I do think it's worth stating that the co-mixture its growth rate seems more similar to that of the delta CCM than of the wild type. And a more clear image of this is shown by the pictures of the cultures at the end of this time. So you can see the delta CCM has grown really well since dilution, and the co-cultures look quite similar to it. While the wild type is not bleaching like before, but it has grown very little since dilution. So this suggests that because the co-cultures have grown and the wild type hasn't, any growth from the co-cultures or most of the growth from the co-cultures is a result of the delta CCM alone, which would suggest that the delta CCM now makes up for the majority of the co-cultures. Now we're able to investigate this using fluorescence microscopy. And this is because our strains have been engineered to have a fluorescently labeled rubisco. So in the wild type, which has rubisco concentrated in carboxysomes, you can see a very clear yellow symbol in a tight compartment, and you can identify that cell as a wild type. Whereas the delta CCM also has rubisco, but it's more diffuse throughout, so it's not as clear. And using this fact, we can look at an image of a co-culture, count the number of cells that are just red versus red with bright points of yellow, and figure out how much wild type or how many wild type cells and how many delta CCM cells we have in a given sample. And so we did this for the different points on our growth curve. We selected these 5 points, and what we would expect to see here is that these first 4 points should be around 50% of wild type out of overall. And we expect this because we started the cultures as a 50-50 mix, and we wouldn't expect them to change throughout because they grew at the same rate the whole time. And post-dilution, there wouldn't really be enough time for anything to change yet. However, when you wait a lot longer post-dilution, that's when we expect that the delta CCM has done most of the growth in the co-culture and that the wild type is still lagging behind. And this was, in fact, the case. You can see here at those same time points that the wild type is around 50% throughout with some variation, while at the very end, it drops down to a little under 25%. And so this is a pretty steep drop already, which is quite impressive. But we were wondering if this fitness advantage of the delta CCM could be further emphasized by repeating the oversaturation and dilution more times. So we did exactly that. We kept the cultures that we were working with before and let them oversaturate again and diluted them, and then did the same thing another time. And we took time points right before oversaturation and then right after growth from dilution. And we measured allele frequencies. And what we found was a steady increase with each consecutive sawtooth down to about 8% at the very end. So this is a really big change from the 50-50 culture that we started out with, and it suggests a quite large fitness advantage of not having a carboxysome in high CO2 oversaturated environments, which is really exciting, but we have a lot more we want to figure out. So some things that we hope to keep exploring is what is it that causes the wild type to bleach earlier? There are some things that we can eliminate because we know that it's a cell-specific phenomenon since the fitness advantage was apparent in a co-culture. And And then we also want to look more specifically at the dynamics of the shifting allele frequency. We got good start and endpoints with the fluorescence microscopy, but this summer I've been working on optimizing a genomic DNA extraction from the cyanobacteria and designing some diagnostic primers so that we can use these in a qPCR to understand how the allele frequencies shift at every point along the growth curve. And finally, we're interested in seeing if this pattern is consistent across other cyanobacteria. So we'll have another strain coming soon that we will be checking the same things out with, and I'm very excited. I'd really like to thank the Lab for having me this summer. It's been great working with Evan and Purva. I'd also like to thank the Berlico Lab for all of their questions and support. Yeah, thank you so much.
Do you have any hypothesis as to why the wild type increased slightly right after deletion in your first assumption?
Let me check. This one?
Yeah, or do you think that's just variance from the deletion?
I think, oh, you're saying that it's higher. I would guess that it's just variance from the dilution. I will say the spectrophotometer we uses, the wild type naturally reads a little bit higher than the delta CCM, even at the same cell counts, because of the way that the carboxysomes scatter the light. So actually, we took, samples and found OD values using our spectrophotometer and cell counts using hemocytometry, and we proved that basically, sorry, it went all the way to the end. Like these three different points that look quite different here are basically significantly, or there's no significant difference between them. So I would be willing to bet that down here, there's also no significant difference between the actual cell counts. It's just how they're measured by the spectrophotometer.
Would you take any fluorescence spectra of these cultures besides just visualization of the colony file for this leaching versus CCM or delta CCM growing?
Yeah, so we haven't yet, but we are planning on it. It's one of the key things in this bleaching is looking at how the phycobilirubin amounts change throughout this time. So that is something that we're working on. And I believe we've used OD 680 in the past to look at loss of chlorophyll, but I didn't do that for any of the samples that I took. So thank you so much.